We're going into a more focused session now where we're going to talk about CTD mutations in particular and characterizing them. And Jonathan is going to come back as a panelist this time for an ACD collaboration as part of the ACD fellowships. So I think many of us parents know that our children have different mutations. There's not a lot of science in knowing what each of these mutations mean in terms of phenotype, and even more so from a biological point, characterizing what they do functionally.
So my name is Peter Axerio-Cilies. I work at UBC in Vancouver. I work with Sylvia Stockler, and this, like Laura said, is a collaboration with Jonathan and Charles at the University of Indiana. So the outline I'll briefly discuss. I know it's already talked about, just a general overview, and then what we're doing in terms of drug screening and developing new therapies. CCD is, it's a disease caused mainly by mutations in the SOC6A gene. It's also caused by deficiencies in the enzymes that make creatine. So it's mostly boys are affected, and there's some female carriers. And the clinical presentation is a global developmental delay, intellectual disability, speech delay, autism, behavioral problems, seizures, GI problems, and low muscle mass. So 3% of the population have this developmental delay. And how to identify this CRTD is mainly by low brain creatine, MRS, and high urine creatine excretion. And doctors prescribe the whole exome sequencing to identify these type of mutations in the gene. So the common treatments are high concentration creatine. Creatine analogs have been tested in terms of like cyclocreatine. The idea of using nanoparticles, but they haven't been used in terms of all the drugs. It's not a common thing. Enhanced creatine synthesis in the brain, as mentioned before, arginine, glycine can be administered. And the idea of using gene therapies, but it is an expensive technology. So what we're trying to do is we're trying to increase, as mentioned, increase the function of this protein, or increase the expression of the protein to the surface. And we're doing this by repurposing drugs that already exist, and screen them virtually initially, and then testing them in the lab. So so what we're doing is we're doing a multi-stage process of trying to find drugs for this. So stage one is to make these mutations in constructs, and then transfect them into, for us, it's hex cells, and then classify them in terms of what kind of dysfunction do they have. So it's partial dysfunction, completely dysfunctional, or hyperfunctional. And then we can quantify this in terms of how much dysfunction there is. Is there, we can actually give a percentage. And then stage two, we use modeling, 3D structure modeling, to identify possible binding sites that can accommodate drugs and possibly increase the function, or help it to fold better, or get it to the surface. And then stage three would be obviously drug testing and dose response curves and binding assays. And then stage four is to identify whether the drug actually gets to the brain, and how it in vivo dose response. And if those conditions are met, then we can consider an N01 clinical trial study. Okay, so the first stage is mainly structural modeling. And then we can actually be able to create a 3D model of this protein. And we try to use it to identify sites on there that can be targeted. This could be even the binding site, the creatine binding site itself, or other sites along around the mutation site. So there are different databases that can be used. We're focused mainly on FDA-approved libraries, which includes mainly drugs that have been approved, but also drugs that are natural products. So this library has been curated, and they also been flagged for example, for toxology, safety, is it taken orally, those sort of things. So we identify sites on the protein, and obviously these sites are characterized by how druggable they are. So in terms of, can it accommodate drugs? Where's the site? Where's the mutation with respect to these sites? And we try to target as many sites as possible and then test them in the lab. So we do some kind of virtual screen. So this is sort of the representation of what happens. So we have a database. These drugs are bound to the receptor or to the transporter, and then they are scored. They're scored based on how strongly they interact to the protein and what kind of interactions are there. And it also tells us the orientation of the drug and how to later on modify it so that we can find drugs that can be more potent. So once we find a drug that we like virtually, then we can buy these drugs and then test them in the lab. So proof of principle initially was, there's two methods that we use. We use carbon-13 creatine labeled and use mass spec to identify how much creatine is actually getting inside the cell. And then method number two is using another methodology, which is electrophysiology, which is a method in which you can measure the currents that are on the cell. And this is caused by the ions going in and out because of creatine binding to the receptor. And this, you can measure it. So we also use that. So the initial proof of principle was to use a variant of a patient variant and then test it and measure the % activity. And here we show that for this particular variant, it was null effect, so no activity for the variant. And then we screen these drugs and identify drugs that can promote the activity of the receptor itself. So we found one compound A, at a certain concentration, almost rescued it completely back to normal. But this was a high amount of drug, so we're trying to find analogs of this drug that can be more potent. So how do we prioritize what type of variants? There's a lot of variants, as mentioned before. So just overall, we data mined on the LOVD database. There was 170 variants. 75 of them were pathogenic, and most of them, as mentioned, were single base pair deletions or missense. Most of the variants observed in single patients, and only 6 observed in greater than 10 patients. So once we created this database, then we can construct them in the lab. So we have plasmids for all of these and more. And so we take these, transfect them in hex cells, and then test them in our assays. So the next step was to create a model for these constructs and try to see what differences, how these mutations may affect the protein, and then use that as a template to screen drugs, particularly looking at the active site, but also looking at where these mutations are on the protein. So the first day was diagnostic testing. So this was just a handful of mutations that we tested, and some had residual activity, some had 0. And this is sort of, we use this as a baseline so that when we test them with drugs, that we know where we are at in terms of the baseline. So we're testing this in both methodologies, electrophysiology and carbon-13, and then try to see how these drugs are being, how they affect all these mutations. So like I said, we use this curated database, and we dock them into the models that we created. So I tried to create both the outward and inward conformations of the protein, and try to see which ones are favorable and which ones are unfavorable. And basically, once we have all the compounds docked and scored, then they're run on molecular dynamics for the top 200 compounds to get more accurate results. And then they're purchased, about 90 compounds were purchased for this initial screen. So this is the type of thing that I see on my computer. It's basically, I look at, there's many things to consider, but the most important are looking at the interactions of how the compound is actually interacting with the protein, scoring how strong those interactions are, what is the orientation of the drug with respect to the nearby residues, and how looking at the drug itself in terms of, is it orally, is there any features on the drug that are toxic? Those are kind of all the considerations that I go through when I choose these drugs. And I make sure that the drug, when I look at how it binds, I make sure that it's binding in a crevice or a concave binding site. And once all that is kind of computed, then I choose the best, 90, in this case, 90 compounds to purchase. This is another one, a little bit bigger. So I try to choose a unique subset of drugs so that we can get a higher probability of success. And this one as well. So all the interactions, we're looking at hydrophobic, hydrophilic interactions, and seeing what the best drugs can fit into a particular pocket.
Peter, quick note that we're at the 2-minute limit, okay. And yes, we absolutely want to see that.
Sure. So this is the drug screening. Basically, we found some hits. So these are the ones that are above the dotted line are increased activity, and the ones on the below the dotted line are inhibitors. So these were the initial ones that were hit rate, hit compounds. We're going to find similar ones that possibly will increase it even further. I've given these drugs to the Schlebach group, so he can test whether these drugs can increase the expression to the surface, and see whether both can increase and promote cell surface. I've also tested butanes in the literature recently and tried to see if butane could actually increase the creatine influx. It showed that it did not. They mentioned also that in their concoction there was cyclocreatine, so possibly cyclocreatine was the result of that positive test, but butane itself does not increase the influx of creatine. So future directions is that the diagnosis, we're going to increase the amount of mutations and test them to see which ones have residual or no activity. Then we're going to test more, screen more compounds, get more sophisticated compounds based on what I really got, and test all the drugs and identify drugs that can modulate the transporter even further. And the ultimate goal would be to find both a drug that can increase the function and increase the expression of creatine, and can be taken orally, obviously get into the brain, and a high potency, because that's in vivo, it gets tough if it's millimolar range. So I'd like to thank the ACD for funding this, and Jonathan and Charles for the collaboration, and Sylvia and Laura and Sangeeta for their support.
Thanks, Peter. So I'll take a moderate the liberty here while Chuck is setting up. So if you go back to your results slide, you're showing one mutation, right? The Pro54LU, right? So you're saying that for one mutation, you're seeing this hits, or are you seeing it for other mutations too?
I'm seeing for other mutations too. So
okay.
we've tested
and
one other
we'll
mutation.
see some of those results.
Yeah.
Okay, and I'm sure we're going to get back to this in the questions. Do you have patient samples for these mutations? This is important for our community because, again, we should make sure that there's a trace back to patients, right? This is not just lovely, it's you have some, okay, thanks. Chuck. So this was a very symbiotic work that we loved having Peter and Sylvia and Jonathan and Chuck work together. And the goal here is to get that, that kickstart of a collaboration that hopefully leads to a lot more funding, like an R1 grant or serious investments. So Chuck, if you want to go ahead and share your screen.
Sure, I'm ready to share whenever I think.
Go for it.
Stop share. Okay. So can you see my screen?
We can. Wonderful. Thank you.
Excellent. Okay, so thank you to the ACD for inviting me to give this talk, and thanks to Peter. So as some of the talks this morning, like Alana and Claire and Laura were talking about, so we're interested in the Schlebach lab on membrane protein misfolding, and we got interested in the creatine transporter deficiency. Is a perfect sort of problem for this. So here you can see some of the locations of some of the missense variants that can cause CTD. There we go. So looking, if you look at this plot on the left, you can see sort of a graph of creatine uptake versus plasma membrane, expression. So this is sort of a way to sort of classify variants based on their effect on uptake versus their, their effect on expression, and whether uptake itself, is affected primarily by retention in the ER. And so we can correct for these things, as Laura was talking about, with correctors. And so let me just minimize this, get it out of my way. Sorry. So, the little yellow dots here are correctors, and they can basically bind to, a misfolded form of the protein in the ER and actually increase, increase the representation of the native folded form of the protein. And so, when we think about the virtual screen, the aim of this work is to sort of expand upon and improve the virtual screen that Peter was talking about. And so, with these virtual screens, you test thousands or even sometimes millions of compounds, and you can tell whether they bind or not. But the goal here really is to increase the hit rate of compounds that we're going to spend time in the lab characterizing, because that is sort of a rate limiting step. And once we get a good pharmacophore using an approach like this, we can sort of do structure activity relationships and sort of rationalize, the binding of, the ligand to either the outward facing or inward facing state. We can really use our knowledge of the transporter in this case to really improve upon this. So Peter talked a little bit about the outward facing and inward facing conformation of the creatine transporter, but I'm just going to go into it a little bit more. So, normally the creatine transporter, the way it works to get creatine into the cells, it really cycles back and forth between these two states. This is the alternating access model. And this is really driven energetically by a sodium gradient. So you'll have the substrate binding to this cavity in between, down here in the center when it's in the outward facing state in the presence of the sodium gradient at the plasma membrane. And there's a conformational transition to sort of an included model where a thin aromatic gate sort of closes over the substrate. And then after that, you have release of the substrate into the plasma membrane, into the cytoplasm upon adoption of the inward facing conformation. Now, the key thing to keep in mind here is that this is a cycle where, when substrates bind, they really favorably go back and forth between these two states. And that's because substrates presumably have very similar affinities for both the outward facing conformation and the inward facing conformation. But you can lock this up with inhibitors. So the way inhibitors work, and this is really important in monoamine transporter pharmacology, is that, in the outward facing state, for example, an inhibitor will come into this substrate site and really lock it into that outward facing conformation, which disrupts this cycle. And it prevents the substrate, in the case of the monoamine transporters, they're neurotransmitters, from being imported into the cell. And in the course of looking for a pharmacological corrector or an activator for the creatine transporter, we don't want to lock the transporter in one of these two states, especially in the outward facing state, because that seems to be the state that is more highly populated in the presence of a sodium gradient. But thanks to work from Michael Friesmith, we know that the folding pathway for the creatine transporter probably proceeds from the inward facing conformation. In the absence of a sodium gradient in the endoplasmic reticulum, when the protein is synthesized, the inward facing state is more commonly observed. And so presumably, if we can differentiate between compounds that preferentially bind to the outward facing conformation and the inward facing conformation, that'll really help us get better compounds and a better hit rate for our screen. And so looking at this, you can really see this, what I'm talking about here. So normally with proteins, you see these ribbon diagrams. The green is the outward facing state and the blue is the inward facing state. But if we sort of look at a surface model of this and sort of cut away through, you can really see the substrate site is roughly in the center of the protein in both, but the overall contacts that substrate makes are going to be different between these two states. And we really want to utilize this in identifying compounds that preferentially bind preferably to the inward facing state. And so we developed a sort of counter screen in the Schlebach lab, in addition to some of the experimental work that we do, in which we dock these candidates of interest to both the outward facing and inward facing compound. And we utilize, there are many different ways of estimating binding free energy. Many of them are very computationally intensive, but there are now neural network based approaches like KDeep, where we can get fast and relatively accurate estimations of binding free energy. And then we can basically calculate the difference in binding energy between the outward facing and inward facing conformation, and then look for biases. So in the case of a substrate like creatine, or another substrate that was identified in the 90s, this ATPCA, you can see from our models that we have relatively minimal biases. So the delta delta G between the outward facing state and the inward facing state are pretty small. But then we also know that people in Randy Blakely's lab back in the 90s identified a bunch of metabolites, some of which are very familiar to us here at this symposium. Overall, we can see that there's a general pharmacophore that we see among many of these metabolites. And that's because creatine itself has this guanidino group that's probably bound within the substrate site to a cysteine residue, and this carboxylic acid group that's bound to either the sodiums or the backbone of TM1. And these inhibitors all have these two functional groups, and we basically want to see if these potent inhibitors have a bias. And so you can look at 3-guanidinopropionate. We know that it has an 84% inhibition. We can see that it has a positive delta delta G, meaning that there's sort of an outward facing bias in these compounds. Same with 4-guanidinobutyrate, it has 38% inhibition. We also measure a very positive delta delta G of preferring the outward facing conformation. So we look at all those metabolites that were identified by Blakely, and you can see, you classify them as in terms of whether they exhibit an outward facing bias or an inward facing bias. We can see that most of the ones with an outward facing bias have lower levels of creatine transport compared to the ones with the inward facing bias. This is just sort of proof of principle that this counter screen would be good at, we want to avoid characterizing experimentally the compounds that seem to have an inward face or an outward facing bias, because these are going to be the ones that really inhibit the transporter. And so looking at the inhibitors that Peter was talking about, we have this candidate 4 and candidate 5. They also seem to display outward facing biases. Then there are a number of activators that were identified, and they're sort of all over the map, which is sort of good. So, we would probably avoid looking maybe at candidate 1 if we were just using the screen, but we would still be looking at candidate 2 and candidate 3. So going forward, we want to dock compounds from our virtual screen in both the inward-facing and outward-facing conformations to help predict the pharmacological effects of top hits. We want to prioritize inward-facing biased compounds to help avoid the discovery of inhibitors. And going forward, the first thing we want to do is refine our inward-facing model. Our original inward-facing model was built on a bacterial homolog that was in the inward-facing state. But there are a million homologs now available. Just this year, I think there was a glycine transporter structure published, and we've looked at AlphaFold for this, but AlphaFold doesn't really seem to differentiate between the inward-facing and outward-facing conformation of the transporter. And if you look really closely at what AlphaFold is doing, it's basically a way of getting automated, very high-quality homology models, which is what we're doing anyway. So we want to repeat the screen on the inward-facing model to help identify new compounds. We want to evaluate the inward-facing to outward-facing bias in relation to the effects of compounds on activity versus expression, and this can help us to specifically identify correctors. We want to also build mutant models of all these compounds, and then also look at this inward-facing to outward-facing bias to see if there are differences with some of these mutants. So shifting gears a little bit, most of the discussion above applies to missense variants. We also know that many of the people in this audience are very interested in nonsense variants. So nonsense variants, a missense variant is basically a point mutation to the transporter that causes misfolding or causes, some direct impact on function, maybe by, existing in a binding site. Whereas these are, normally in the process of protein synthesis, you have a start codon, and then you have some stop codon at the end, and in between you have an open reading frame. The ribosome encounters the start codon, begins translating the mRNA, and you get some, synthesized nascent chain out of it, which begins to fold. With, in many cases, there are single nucleotide transitions that can occur that can turn a certain codon into a stop codon. And when that happens, you begin to synthesize the nascent chain, and then it encounters this premature stop codon, and the truncated protein comes out, is usually degraded, and then the mRNA is degraded by a process called nonsense-mediated decay. So people have thought about ways to address these mutations by tricking the ribosome into basically skipping over these premature termination codons and reading through the message to synthesize a whole protein. And people have done this in the past using antibiotics like gentamicin and G418. But in the, I think, mid to late aughts, this atelorin was identified. It was actually approved for use in Duchenne muscular dystrophy. So we were interested to see if any of these compounds would have an effect on nonsense variants for the creatine transporter. And long story short, you can see that for the antibiotics, they have these really big error bars, and that's sort of, antibiotics are very toxic to mammalian cells and eukaryotic cells generally. And that's why we get this sort of signal. But if you look at atelorin, particularly in the case of this arginine 514 termination, you actually see a measurable increase in expression. So the next thing we want to do after this is move forward, and Peter's going to, see if the effect on function is, see if function is rescued in these, but we're seeing a measurable impact on expression in the Schlebach lab. And that's actually a really promising note to end on. I want to thank everyone in the Schlebach lab who's worked on this project. And I also want to thank our collaborators at the Simons Foundation of the University of British Columbia, Sylvia and Peter. It's been a very fruitful collaboration. I'm very excited about what we can do going forward. And last but not least, I want to thank the ACD for their support of this research. And it's been really great to see some of these talks here today. And with that, I'm looking forward to the Q&A.
Thanks so much, Chuck. This was great. It was lovely to see the results, and I think you addressed beautifully some of the questions from the parent side, including nonsense mutations and how are we thinking about that. So I want to point out that drug repurposing is a very promising area that usually, and it's coming to focus more recently, but it's not, it hasn't scaled so far. So what we're trying to do here is invest in areas that have a lot of promise, and this is what these collaborations are showing. So on that note, I'll invite our moderators for the session. So Franzisca Haderk is a researcher at University of San Francisco, and she comes from a different background, and we're excited to have her as a volunteer in the ACD world. And Erin Coller is our director of communications, and you've seen her live tweets of the conference throughout. And Dr. Sylvia Stockler is a longtime member of the ACD and part of this research collaboration. And Jonathan, you already know, he's a relatively new member of the ACD community, but he's been doing a lot of good work for CCDS. So Francie, Erin, you can take it away and take turns with the questions.
Yeah, thank you so much for the kind introduction, Laura, and thank you for all the speakers for their great talks. It's really great to see the science moving forward. I think my first question, going chronologically, is to Lara. Very great work also moving into in vivo and PBPK analysis. As we were looking more at the chaperone targeting drugs or drugs that increase stability of SLC6A8, what are in general side effects and how do you explain side effects in this area?
Yeah, so I think the side effects that we were seeing, we didn't go into too deeply because we saw them at doses that we knew were going to be efficacious. So we didn't have a therapeutic window, like I mentioned, unfortunately. The side effects were general physiological changes, nothing like really pointing to something specific from like a corrector approach, for example. So I would think that the side effects could be mitigated through more complex medicinal chemistry and maybe a very more like tailored dosing regimen. But I don't think it's necessarily applied to correctors in general. I think it was more for the molecule itself.
Yeah, just a follow-up question also. You have in your very first slide, I think you have shown a nice plot of like the different mutations that can occur, and that is very diverse mutational categorization. When you have a mutation that would occur early in early exons and would truncate the protein significantly, would you think that increasing stability of the protein is enough for shuttling some level of creatine, or is there certain mutations where targeting protein stability in general is better?
Yeah, I see what you're saying. I think there are, like from that first slide, I was trying to paint that same picture that has come up in a lot of different talks today, that there are different categories of these mutations. And I think for a corrector, like thinking how Vertex developed therapeutics for CFTR, there were correctors that worked on some variants because all you needed was to get it to the surface. Then there were potentiator molecules, which getting it to the surface wasn't enough, and you really needed to activate the protein that was completely dead when it was at the surface. So I think like a similar approach would be needed for CTD, and it wouldn't be one per mutation. Maybe you could just categorize it in these buckets and see like correctors will get it to the surface, which ones can work just with that treatment alone, and then which would need a follow-up potentiator to activate. And I think for some of those early truncation mutations, maybe you even if you get it there, there's not enough of a channel to even transport. So some might be excluded from both of those approaches. But there are, like I said, almost 70% have there's so many mutations that have a lot of protein retained. So I think that would cover a really wide portion of the population, and then other approaches like gene replacement or things would be needed for those that don't have any protein.
Thank you so much. We have quite a few questions coming in for Peter and Chuck as well. Peter, can you tell us how your team is thinking about moving these findings to patients and expand at all if possible on the timeline and how long it might take to get to those N-of-1 trials?
Okay, so because we're focusing on FDA-approved drugs, and I've been mainly focused on drugs that can be taken orally and safely, is if the drug, it really depends on the drug itself. So, and obviously the biology. So it's very easy to pick a drug that's very safe and everything, and then you test it, it doesn't work. And there are times that drugs are very complex but still work. But the methodology, what we're doing is we're trying to find something, and our first stage is trying to find something that really works and really works for one particular mutation, or perhaps even 2 or 3 mutations. So if those drugs can be taken, and obviously by working with Dr. Stockler, she can tell me, is this something that can be taken safely, and then you also have that information already. If the drug's already been published, you kind of know what kind of dosing has been used in the past. Is that dosing makes sense for a patient? So these are the type of questions that we have to ask prior to giving it to someone. So the answer is yes, it could happen that the drugs, if we test them, they work and it's safe, and they may not even, if it's safe and we get it to the dose that we test in our assays, can go to the brain at those concentrations, then I could see it that we can move pretty fast and get it to a patient. But it has to meet these criteria that it has to be effective in the lab, and then does it work in terms of the concentrations in the lab? Does it get into the brain at those concentrations? And is it safe? So if it meets all those criteria, then I could see it happening very quickly. If the drug is more, hasn't been studied that well, or we don't know how much of it actually gets in the brain, that's an extra step that we have to do, in animals and before we go into people. So it's hard to say, but there is a path to getting it quite quickly if the drug is safe, and that's the most important thing.
Can you share a bit, for our parent community is probably wondering, what does quickly mean? How long?
Really, it's like,
are we talking months, years?
I would say, it could happen in a year. It could happen that a drug, it's hard to say, pinpoint a specific day, but I could see in a year that we can come up with something, a drug that is safe. It meets, it works in every single condition, and then it can be taken to a patient. So I would say a year would be possible.
Thank you so much. We had one more question for you here. How many of the drugs worked on more than one kind of mutation?
So, from like the 8 that worked, I think 6 of them worked on other mutations. So I tested in one other mutation, and there was 2 of them didn't, but the other 6 did. So at least in six drugs, it worked in 2 mutations. So we're going to take those 6 and test them in the whole mutations that we do have, and see if it's it works in every single mutation. But yes.
From Chuck's talks afterwards, is there, because of the inward facing or like preference, is there a benefit for reversible drug binding versus constitutively binding? Could you explain this again, like how you need this cycling and how drugs?
Oh, you're talking about like drugs like ATPCA that can bind to both. We see this in monoamine transporters too, like things like amphetamine that can really promote flux either way. I think that the evidence seems to be that as long as it can bind to the inward-facing conformation, then it could potentially be used as a corrector. Now, that's not to say that all things that bind and stabilize the outward. Anytime you stabilize a conformation of one of these things, you're probably going to promote, good folding. But as a drug treatment, you probably don't want something that's inhibiting your target, right? So that's sort of the double-edged sword that you have to navigate.
But would you opt for constitutively binding, or is it all a reversible binding?
It would be interesting to explore that. I mean, our screen doesn't really look for constitutive binding, but it's certainly something that we could look at.
Mm-hmm.
I mean, we know that, for example, in the case of rhodopsin, we know that retinol binds binds constitutively, and it certainly stabilizes rhodopsin. As long as it doesn't impede the transport cycle.
Yeah.
That's a promising avenue.
would like to ask one more question, maybe towards Lara, because you have also explored, you have shown different creatine levels, including in male, as well as in also heterozygous females. And I think the conclusion there was that in heterozygous females, you have average slightly higher creatine levels and a more moderate phenotype. Do you think it's really just the creatine levels, or are there other like feedback mechanisms? And yeah, how much have you explored the female CTD phenotype?
So we haven't explored it too much, but I think what you said is exactly right, that because there's one wild type copy there, there's going to be a more creatine in more cells and also a more mild phenotype because of that. And I think there's also evidence for that same spectrum based on all the different patient mutations. When you have a huge, like if you have a protein that has no transport, like the delta F5408 mutation, you're going to have almost no like creatine in their brain, have a more severe phenotype. So I think there is that spectrum. For the females, one other thing to keep in mind though, is that there's exon activation in the brain of animals. And so not just because like you could have 10 females that are carriers, not all of them are going to have that wild type X copy on in every cell or the mutant. So I think there's going to be a mosaic phenotype even within one person. So I think that has to be kept in mind as well when thinking about like the moderate phenotype in the females.
Mm-hmm.
And Peter, you mentioned that you also curated FDA-approved libraries in your work. Did any of these make it to your short list of about 90 compounds? And is there a certain number of them that were considered safe, and which ones?
Yeah, so the curated list incorporates everything, all the FDA-approved drugs, natural products. And then I went through with, done this in the past, where I went down to the list and sort of, made a list of oral, IV, intranasal, all that. And then safety is, tolerance, like what doses have been used in the past. And so when I do the screen, you end up having, a mixture of drugs that are on the top of the list that may be, safe, relatively safe, or toxic. But I try to choose compounds that strictly are orally and relatively safe or relatively used frequently. So on that list, I tend to choose ones that are safe already. So we test in the lab stuff that are safe and tolerable. It's quite useless to, unless I really, and there might be times where I make exceptions if the science is telling me that choose the toxic one initially, I would, but 95% of the time I pick safety. So they'll always be on the top of the list. And when we test them, they'll be, safe, considered safe. So the answer is yes, it's always in there.
And did those make it to that short list?
Yeah.
Okay. Also, can you hypothesize how much creatine uptake needs to be rescued to achieve behavioral correction? For instance, 20% or 80% of wild type?
That's a good question. So I don't know the answer to that, but the idea there is that if we can test it in essentially, it would be nice to do it in animals, so that if we have like 80%, like you say, and then test it in an animal and seeing that there is increased creatine levels, then that's a good experiment to satisfy that. We haven't done that yet, but certainly the aim of this project is to get it as high as possible. So if there is a drug that is safe and we're getting 40%, we can test it in an animal and see if that 40% is relevant. If it's not, then we have to increase it to 80. So that's an experiment that can be done relatively quickly if we find that drug.
Is your biomarker always creatine plasma levels, or are you in the experiments also looking into animal, like brain cells in animals?
Brain in the brain. So we can measure like CSF and stuff.
Yes.
For Jonathan or Chuck, we saw from Resolute that there may be other transporters that can transport creatine. Are these other transporters structurally similar to SLC6A8?
Yeah, so Chuck is more of a transporter person than I am, but the SLC6 family is specifically, so, and that's about, I think there are 19 family members. So they mentioned hundreds, and basically ones that are SLC something other than 6 probably have little homology, but the SLC6 family are very similar. That includes a serotonin transporter, norepinephrine, glutamate transporter, a bunch of others, right? I don't know if you want to add to that, Chuck.
Yeah, I will say that their overall three-dimensional architecture, like the backbone locations of where the C alpha carbons, that's relatively conserved, but one promising thing that I initially thought when I thought about this in terms of the inward facing conformation is, well, why don't we see if ibogaine works? We know that ibogaine binds selectively to the inward facing conformation of the serotonin transporter. And that's actually how Michael Friesmith first came up with some of the hypotheses about that being the default state in the ER. Then I looked at our homology model, and the residues lining the inward facing permeation pathway, and they're just different. So I would suspect that ibogaine isn't going to work. So, I guess what I need to say about this is that we can use other SLC6 transporters as in a drug screen. And we could, for example, the glycine transporter, which is SLC6, we can use that to build better models of the creatine transporter, but the individual identities of the amino acids lining surfaces, that's going to matter for drug binding.
But yeah, more generally though, this approach should work for any of the other 19 transporters. So if there are other transporters of interest, we could hypothetically apply this just by building slightly different models. And I should also mention too, I don't know if Laura, if Janana is doing any more lead compound optimization, but this could also be used to help guide SAR. If anybody wants to make next gen compounds, we could at least help maybe screen out some of the modifications that are not so good.
So I know we are almost out of time and we're supposed to go for a break, but if you might indulge me. So I know we've compared the structural model of SLC6A to the serotonin transporter, but that transports a fundamentally different substrate, right? If you were to look at the structure of the creatine binding pocket of another transporter that transports creatine, do you think, based on your model, if you identify drugs that are actually linked to the creatine binding pocket itself, could those be applied to those other transporters, even if they're not structurally, the rest of them are not similar, but the creatine binding pocket might be? Could you use that then to transport more creatine?
Yeah, I mean, if you demonstrate in an assay that some other SLC6 transporter is able to uptake creatine, it would be very surprising if the contacts, if you looked at the actual substrate binding pocket, there weren't similarities in such cases. In fact, I mean, I'm sure that there would be. And maybe it doesn't do it with as much efficiency, but it probably still works, and you could certainly use that to gain more insights.
Yeah, I think the other point you're getting at is whether we could use the screening approach to answer questions of specificity, which I think we probably could. The question is whether we would have training data, right? The creatine transport stuff we dug out of Blakely stuff allowed us to see empirically whether our approach works. And so if there were data like that, I'd really be interested in looking at this in terms of specificity for one transporter over another. But Chuck will tell you there's a lot of more similarities than there are differences about how these transporters work for creatine versus other, even serotonin, right?
I mean, it's remarkable actually how much overall structural similarity there was when the serotonin transporter structure was first published. Homology models based on, the bacterial homologue were actually really good. There were some differences, but the overall locations of certain residues in the binding pocket were very similar.
Before we wrap up, I think it would be great for our community to hear from Dr. Stockler. So Dr. Stockler, we know that you've been involved in the registry work with ACD and the core outcomes work. So can you comment on how this work will be enabled to get us to N of 1 trials and the importance of core outcomes and the registry, how that all relates?
Yeah, what we're doing in parallel to Peter's and to Chuck's work is to also to get ready in the on the bedside for trying those drugs. And as Peter mentioned, it really depends on what are the properties of the drug, what will be the dosage in which we are going to take it, and what are the issues about giving these drugs to young children, and what are the safety criteria. What we have started now is just on a regulatory perspective to discuss with Health Canada, which is the FDA and EMA equivalent in Canada, to get them involved into the situation that we might be when we do have a drug. So to get their advice how to implement this drug in the clinical setting and how to make sure that we also can give it to young children. That's the regulatory perspective. And then, of course, because these drugs will be mostly targeted to very particular mutations, there will be only very few single patients that can undergo that proof of principle on the clinical side, and that will be with N of 1 trials. And we're working on setting up the methodologies for N of 1 trials together with Beth Potter's group, who is also going to talk today in Ottawa, and with a group in Toronto, to get some ideas about the methodologies, how to conduct the N of 1 trials, so about the trial designs. And we're at the same time also working on legal perspective and ethical perspectives, how to implement this trial. So overall, if it comes to a clinical application, there will be all kind of issues that we have to consider before we start those trials, and they will be started in single patients only. And of course, we need the registry for understanding what kind of outcomes we're going to measure. It was already mentioned, how can we make sure that we can have a change in behavioral outcomes? What would be the creatine concentration in the brain to have those behavioral outcomes? But the most important thing is to know how to measure and what to measure in terms of behavioral outcomes. And for this, we need the registry, which gives us a good understanding of the natural history and also the input of the patients themselves, which gives us an idea about outcomes that are important for the patients.
Thanks,
so it's
Sylvia.
a whole
this was
different
great.
type of research, and so I hope that we will be ready when Peter and Chuck can kind of tell us which drug we should use.
Yeah, we have to push on all levels, but this was a great way to kind of like wrap up the section. So I want to thank our speakers and our moderators again. I think we've seen some great work on drug repurposing, and we're going to transition in the next session in understanding more about the patient registry and newborn screening.